Eukaryotic RNA polymerase requires general transcription factors. The initial finding that, unlike bacterial RNA polymerase, purified eukaryotic RNA polymerase II could not on its own initiate transcription in vitro led to the discovery and purification of the general transcription factors. These accessory proteins assemble on the promoter, where they position the RNA polymerase, pull apart the double helix to expose the template strand, and launch the RNA polymerase to begin transcribing. Figure 7.12 shows how the general transcription factors assemble at a promoter used by RNA polymerase II. The assembly process typically begins with the binding of the general transcription factor, TFIID, to a short double helical DNA sequence primarily composed of T and A nucleotides. Because of its composition, this sequence is known as the Tata sequence, or Tata box. Upon binding to DNA, TFIID causes a dramatic local distortion in the DNA, Figure 7.13, which helps to serve as a landmark for the subsequent assembly of other proteins at the promoter. The Tata box is a key component of many promoters used by RNA polymerase II, and it is typically located 25 nucleotides upstream from the transcription start site. Once the first general transcription factor has bound to its DNA site, the other factors are assembled, along with RNA polymerase II, to form a complete transcription initiation complex. Although Figure 7.12 shows the general transcription factors piling onto the promoter in a certain order, the actual order of assembly probably differs from one promoter to the next in the cell. After RNA polymerase II has been tethered to the promoter DNA in the transcription initiation complex, it must be released from the complex of transcription factors to begin its task of making an RNA molecule. A key step in this release is the addition of phosphate groups to the tail of RNA polymerase, an act performed by the general transcription factor TFIIH, which contains a protein kinase enzyme as one of its subunits. See Figure 7.12e. This phosphorylation is thought to help the polymerase disengage from the cluster of transcription factors, allowing transcription to commence. Once transcription has begun, most of the general transcription factors are released from the DNA so that they are available to initiate another round of transcription with a new RNA polymerase molecule. When RNA polymerase II finishes transcribing, it is released from the DNA. The phosphates on its tail are stripped off by phosphatases, and it can reinitiate transcription. Only the dephosphorylated form of RNA polymerase II can initiate RNA synthesis at a promoter. Eukaryotic RNAs are transcribed and processed simultaneously in the nucleus. Although the templating principle by which DNA is transcribed into RNA is the same in all organisms, the way in which the RNA transcripts are handled before they can be used by the cell differs greatly between bacteria and eukaryotes. Bacterial DNA lies directly exposed to the cytoplasm, which contains the ribosomes on which protein synthesis takes place. As mRNA molecules in bacteria are transcribed, ribosomes immediately attach to the free 5' end of the RNA transcript and protein synthesis starts. In eukaryotic cells, by contrast, DNA is enclosed within the nucleus. Transcription takes place in the nucleus, but protein synthesis takes place on ribosomes in the cytoplasm. So, before a eukaryotic mRNA can be translated, it must be transported out of the nucleus through small pores in the nuclear envelope. Figure 7.14. Before a eukaryotic RNA exits the nucleus, however, it must go through several different RNA processing steps. These reactions take place as the RNA is being transcribed. The enzymes responsible for RNA processing ride on the tail of the eukaryotic RNA polymerase as it transcribes an RNA, processing the transcript as it emerges from the RNA polymerase. Figure 7.15. Depending on which type of RNA is being produced, an mRNA or some other type, the transcripts are processed in various ways before leaving the nucleus. Two processing steps that occur only on transcripts destined to become mRNA molecules are capping and polyadenylation. Figure 7.16. 1. RNA capping involves a modification of the 5' end of the mRNA transcript, the end that is synthesized first during transcription. The RNA is capped by the addition of an atypical nucleotide, a guanine G nucleotide with a methyl group attached. This capping occurs when the RNA polymerase has produced about 25 nucleotides of RNA, long before it has completed transcribing the whole gene. 2. Polyadenylation provides newly transcribed mRNAs with a special structure at their 3' ends. In contrast with bacteria, where the 3' end of an mRNA is simply the end of the chain synthesized by the RNA polymerase, the 3' end of eukaryotic RNAs are first trimmed by an enzyme that cuts the RNA chain at a particular sequence of nucleotides, and are then finished off by a second enzyme that adds a series of repeated adenine A nucleotides to the cut end. This poly A tail is generally a few hundred nucleotides long. These two modifications, capping and polyadenylation, are thought to increase the stability of the eukaryotic mRNA molecule, to aid its export from the nucleus to the cytoplasm, and generally to identify the RNA molecule as an mRNA. They are also used by the protein synthesis machinery to make sure that both ends of the mRNA are present and that the message is therefore completed before protein synthesis begins. Eukaryotic genes are interrupted by non-coding sequences. Most eukaryotic RNAs have to undergo an additional processing step before they are functional. This step involves a far more radical modification of the primary RNA transcript than capping or polyadenylation, and it is the consequence of a surprising feature of eukaryotic gene arrangement. In bacteria, most proteins are encoded by an uninterrupted stretch of DNA sequence that is transcribed into an RNA that, without any further processing, can serve as an mRNA. Most eukaryotic genes, in contrast, have their coding sequences interrupted by long, non-coding intervening sequences, called introns. Figure 7.17. The scattered pieces of coding sequences, or expressed sequences, called exons, are usually shorter than the introns, and the coding portion of a eukaryotic gene is often only a small fraction of the total length of the gene.